During this lesson, we will investigate mitosis, a type of cell division, and the cell cycle. We're going to hit a few vocab terms first. So, chromosomes are thread-like pieces of DNA. In the human cells, we have 23 types, or 46 total. And it's important for you guys to realize that chromosomes come in several shapes and sizes. Chromosomes can be made of chromatids, which are just the copies of the chromosome. So sometimes we have chromosomes that are made of a single copy or chromatid. And so here we have a chromosome made of one chromatid. Cells are going to go through replication, at which time they're going to make a copy of that chromosome or chromatid. So after replication, we're going to have a chromosome made of two chromatids. Those are exact copies of each other, and they're going to be connected in the center by a centromere. So just keep in mind, as long as those two pieces, those two sister chromatids, are connected in the middle, they are a single chromosome. So one structure, one chromosome. So here's another picture of our centromere. This is a protein structure right in the middle. It's going to hold these two sister chromatids together. Now these sister chromatids are the two pieces of our X-shaped chromosome, and they are exact replicates of each other. So two identical copies of the DNA. This is important because during mitosis, during cell division, we're going to split those two copies up and divide them into separate cells so that we get two identical daughter cells. Okay, so remember, chromosomes can have a single copy, be one thread-like structure. After replication, we'll have a X shape, so two sister chromatids connected at a centromere. And remember, DNA can condense and get very thick into these nice X shapes when the cell is ready to divide. A few more vocab terms. Centriole are pieces of cytoskeleton that produce spindles. And the spindles are going to be the fibers that connect to our chromosomes and move them around and pull those sister chromatids apart. If we put the spindles and the centrioles together, we call that a centrosome. So just a tip for keeping these two words separate in your heads. Centrosomes have an S, so they produce spindles. Centromeres have an M, and they're in the middle of chromatids. So the life of a cell is in a cycle. It's like a circle. It starts with a phase called interphase, and that's where the cell spends most of its life. And then we have a shorter period of division called mitosis. To help you guys remember the steps of mitosis, you can think about this silly little saying over here. So I pray more at the church. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. So the two parts of the cell cycle can be broken down into shorter stages or steps. So for interphase, we have gap 1 or G1 phase. This is when the cell goes through some growth and then regular, normal, everyday functions. So a skin cell is going to be protecting you, a heart cell will be pumping blood. The second part of interphase is S phase, and this is when DNA is synthesized or replicated. So S phase is for synthesis. The last part of interphase is G2 or gap 2. This period of growth is preparing the cell for division. So it doubles all of the cytoplasm, increases the number of organelles, and just gets ready to divide. So it's very important that you guys remember this replication of the DNA producing sister chromatids occurs during interphase, before mitosis even gets started. So replication occurs during the S phase, or synthesis phase, of interphase. So mitosis is broken down into many steps, and this is the process of duplicating the nucleus and dividing that genetic material into two separate cells. Cytokinesis, we're going to see, is the process of dividing the actual cytoplasm. So cytokinesis, dividing cytoplasm. At the end of mitosis, we're going to wind up with two daughter cells. So we start with 1, end with 2, and they're going to be identical to each other and identical to that original cell. We're going to divide everything equally here. We're going to copy the DNA, put copies in each cell, we're going to divide the cell guts, and end up with clones, or identical copies of the cell. There are 3 major reasons that cells go through mitosis. The first one has to do with growth. So as a plant grows, we're going to see that they're going through mitosis. And plants tend to grow at places like their root tips. Kids are growing all the time as well, so we see them going through lots and lots of mitosis. Cells also go through mitosis to repair themselves. So if a cell is damaged, say you nick yourself while you're shaving, mitosis is going to replace those damaged cells and heal that wound. Same with this lizard down here. If he loses his tail, we're going to see mitosis occur to replace that lost tail. The third reason cells go through mitosis is asexual reproduction. So some organisms, like this amoeba over here, it's a type of protist, will go through mitosis to divide and produce a copy of itself, or a clone. So there are 3 major reasons for mitosis: growth, repair, and asexual reproduction. The best way to study mitosis is to draw what's going on and then write down the steps and describe what's going on in words as well. I'm going to walk you guys through how to do that. The first part of the cell cycle is interphase. During this time, our cell will have a nucleus, and all of the DNA is going to be loose and stringy and look like chromatin. So we cannot tell the difference between all of our chromosomes. During prophase, the first step of mitosis, we're going to see that our cell has a nucleus that's dissolving. So the nuclear membrane breaks up. The chromosomes are going to condense and show up as these thick X's. Our centrosomes are going to be migrating towards the poles. And those little pieces of cytoskeleton are going to produce spindles as they go. The second step of mitosis is metaphase. The chromosomes are going to line up in the middle of the cell during metaphase. The centrosomes are going to be at the poles, and our spindles are going to connect to the center of our chromosomes. The third step of mitosis is anaphase. During this step, the spindles shorten, the sister chromatids are ripped apart. This means that we have one copy of the DNA going to each side of the cell. Those chromosomes are moving because the spindles are shortening. So at this step, we see that the centrioles are acting kind of like a fishing line. They have cast out their spindles, they caught hold of the chromatids, which are kind of like the fish, and now they're reeling them in, shortening those spindles to pull the chromatids towards them. The last step of mitosis is telophase, and this occurs at the same time as cytokinesis in most cells. Telophase focuses on the nucleus. Cytokinesis is actually the division of the cytoplasm. So during telophase, we see that our nuclear envelope is going to reform on both sides of our cell. The DNA is going to start to uncoil, so it becomes chromatin again. Okay. For cytokinesis, we see a cleavage furrow forming, so this pinching is occurring, which will actually divide the cytoplasm and give us two separate cells. So in that previous slide, we were looking at an animal cell that has a flexible plasma membrane. I want to point out to you guys that when we talk about plant cells, they are surrounded by a thick cell wall. And that cell wall contains a lot of cellulose, so it is rigid and inflexible. Plant cells are not able to pinch the way animal cells do. During telophase, we're going to see the same types of things going on for this plant cell as we do with our animal cell. The nuclei are going to be reforming. The DNA is going to be uncoiling, going back to chromatin. During cytokinesis, we see something a little different. So instead of pinching, this plant cell is going to lay down a cell plate. And eventually this cell plate is going to thicken and become a brand new cell wall dividing the two plant cells. So the products of mitosis are two daughter cells, and these cells are going to be identical to each other and identical to that original parent cell we started with. So I suggest you guys take a minute and practice drawing mitosis and fill in the table with the description that goes with it.